Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, TuneIn Radio, GoodPods. Wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X as Let's Talk Micro 1, LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com where you can send any feedback, any suggestions. So please subscribe to the podcast, download episodes, leave a review if the app allows you to do so. As always, I am grateful for the support and any suggestions, any feedback, they are always welcome and appreciated. And if you haven't checked out the latest episode, please go ahead and do so. It was the second part of a great conversation about emerging viruses with Dr. Ryan Relich and Dr. Benjamin Pinsky. And in this episode, they talk about diagnostics, lab testing, the challenges that they are with developing tests, and they give their views on viruses and outbreaks. So both episodes are great. From the first one was about breaking down DNA viruses, RNA viruses, diseases, mode of entry, and then this last one about testing. So definitely check both of them out if you haven't done so already. It was a very educational episode, and I had a great time recording it. Their chemistry and their passion, it was amazing. So please check it out. So today's episode is one that I have wanted to do for a long time, and I finally, was able to. And it is about veterinary microbiology. So this was a great episode where Kelli Maddock and Sarah Gefroh from they're two medical laboratory scientists that work in a veterinary lab. So they are from the North Dakota State University Veterinary Diagnostic Lab. And they came to the podcast to talk about veterinary micro. We talk about susceptibilities, the CLSI. I asked the question, what led them to choose, working in a veterinary lab. And, Kelli had an interesting answer, which was about that she was experiencing burnout, which is something very unfortunate, but very real that happens in this field, with all the work we get. And sometimes, we're understaffed, the volume. So it's a very unfortunate reality of this profession that I hope that we can alleviate. But it was definitely, it was such a great episode. Seeing, what they're doing, the workup of cultures, talking about the organisms, susceptibilities, and something very interesting that was said in this episode is that if you think about human medicine, right? So if we have some sort of infection, we immediately go to the hospital, pretty much, right? And we are seen and they collect a sample, and then as microbiologists we get that sample is relatively fresh, right? The infection hasn't been going on for a long time, so we get to work them, but with right away. But with veterinary medicine, sometimes the animal has been dead for a couple days, and then the sample is collected. So that is something that they have to account for when they're working cultures. They have some environmental factors, so the organisms that are present, whether they've been there or they got them from the environment. So it has its challenges. But it was definitely a great episode, very educational, and I enjoy recording it, and I hope that you enjoy it as well. So let's go ahead and listen to it. As medical laboratory scientists, we can go to many areas. As we work, we can go to, some of us go into education, some people go to industry. So there are many options. And then maybe you have heard about, maybe what about veterinary, performing as a medical laboratory scientist in a veterinary lab? What about veterinary microbiology? So today with me, I have two guests that are going to be talking about that. With me, I have Kelli Maddock and Sarah Gefroh from the North Dakota State University Veterinary Diagnostic Lab. Kelli and Sarah, welcome to Let's Talk Micro.
Hey, thanks for having us.
Thank you.
My pleasure. So let's go ahead and start with a quick introduction.
I'm Kelli Maddock. I'm section head of the microbiology and biosafety level 3 laboratories at the North Dakota State University Veterinary Diagnostic Lab. Um, do a little bit of everything here. There's a lot of opportunity in veterinary microbiology and veterinary medicine in general for a medical laboratory scientist. My bachelor's is in medical laboratory science, and my master's is as well. I have both human and veterinary experience. And because of that, I feel like I've gained a lot of interest and perspective in One Health and public health. So in 2021, I started my doctorate in public health at the University of South Florida. So currently in progress with that, hoping to graduate in May of 2024.
And my name is Sarah Gefroh. I'm a diagnostic microbiologist here at the Vet Diagnostic Lab. And I guess, like that implies, I work in the microbiology lab. We do parasitology, micro, mycology, all of the things that come with microbiology. I also help Kelli in the biosafety level 3 laboratory as well.
Well, welcome again, and thank you so much for taking the time. It's a pleasure having you both here. And I was talking to Kelli before we started. Like and the audience can't see this, but on the background she has the ASM books. So really, great resources. So definitely, if you're working in this field, I invite you to definitely check them out and get as familiar with them as you can because they are very helpful and they contain a great deal of information. Okay, so before we go on about we start talking about what you do in the lab. So let's start with the first question. So what led you to work in a veterinary lab?
For me personally, I was experiencing burnout and I was at the point of looking at becoming an accountant because there were some free opportunities for extra education there. Loved laboratory work, but I was so burnt out um, working where I was working. Uh, and I met a veterinary microbiologist at a public health biosafety level 3 select agent training. And she encouraged me to watch for open positions. And I was initially hired on at the Veterinary Diagnostic Lab in 2015 as a diagnostic molecular microbiologist. And because I had an interest in pretty much all areas and naturally as a med tech, I am nosy. So asked a lot of questions and was willing to try testing in multiple sections, and it just kind of morphed from there. It this field is perfect for anyone who has lots of questions and has an interest in animals. I think I literally learn something new every day.
And for me, I guess I started in the human realm as well. With my degree in MLS, I worked at a pretty large size microbiology lab for about 15 years, 10 of which I served as an education coordinator for an MLS internship program. And I love my profession and I like to share information about our profession. So that was very rewarding to me. But as the years went on, I found that I also wanted to have an opportunity to learn a different aspect or a different perspective of MLS. So I knew Kelli well, and she encouraged me to give this a try. So about 3 years ago, I came over here to veterinary land, and it's been great. There's always something new to learn, a lot of different aspects that you can incorporate in your day-to-day work that have been very rewarding.
Yeah, that's great to hear. And Kelli, that's an unfortunate thing that happens in this field too. We definitely have a lot of work. We're dealing with issues, right? Unawareness of the profession, staffing, and for the audience, even as MLSs, sometimes in microbiology we have this issue where, let's say, like if you work in hematology or chemistry, right? You work first shift and then you end, and the person that's taking over in the second shift, they have the same training as you do pretty much, so you can just turn over the work. In micro, sometimes the flow it's a lot different, and most of the readers and cultures, they're on first shift, and the person that comes to relieve you on second shift, they don't have the same training. So that means that you're kind of stuck with all your workload for that day before you go home. So very challenging to complete it in 8 hours. It's typically 9, sometimes 10, and that unfortunately it leads to a lot of burnout, and we lose a lot of people. So it's something that we definitely need to address and continue working on. Okay, so as far as I met people in my career that sometimes they had some experience working. Like I met someone that worked for a zoo. I met someone that graduated our program, and then she went on to, after a year or so, to go into SeaWorld. But as far as the education, requirements to work as an MLS in a veterinary lab, they're the same for us. As long as you complete an accredited program for medical laboratory sciences, you're able to work in a veterinary lab. Is that correct?
Well, actually, as a medical laboratory scientist, you are very well suited to make the transition. Minimum requirements. So veterinary laboratories are accredited, but they aren't accredited by like CLIA or CAP. It's the American Association of Veterinary Laboratory Diagnosticians, and they may also look at ISO accreditation, the 17025, depending on what sort of testing they do. So from that AAVLD accreditation requirements, most requirements just indicate that a 4-year degree in some kind of biological or chemical science is required for section heads and other roles. For instance, we work with veterinary pathologists. Obviously, the person should be a veterinarian who is trained as a pathologist and boarded in that respect, or a highly skilled toxicologist who's a veterinarian with additional chemical training or chemistry training. So there are minimum requirements, but usually a 4-year degree would set you up for it. But an MLS is really uniquely poised to just jump in and learn more species. So human medicine, we learned so much about humans and what our normal ranges are and the ins and outs of diagnostic testing. But in animals, you have to consider all of the different animal species. So it isn't just your dogs and cats. You have to think about cattle, small ruminants, exotic animals. We never really know what we're going to see in a single day. So you also have to be aware of the oddball things that you learn about in your training. So a lot to consider. Sarah, would you have anything to add?
Yeah, I think and you can correct me if I'm wrong here, Kelli, too, but by having MLS personnel here on staff, it allowed our veterinary diagnostic lab to then kind of bridge that gap during COVID testing. We were able to, attain a CLIA certification so that we were able to help out with human testing or overflow testing for our state health lab. So while an MLS was not required, it has served as a great benefit for our region in many aspects.
Yes, and for that human COVID testing, we did require medical laboratory scientists because North Dakota is a
licensure
state. Yes, but because we were trained and appropriately licensed, we could jump in and help, and that better serving public health. But other states across the United States where they might not have had those requirements still also jumped in and did help with that overflow testing too.
Okay, and so now that you mentioned the different ranges, and so we know, as right, we complete let's say a medical laboratory sciences program, and any job that we start requires training. So now that you mentioned that, so it's basically we go to school, and then the ranges that we learn, they're more for a human population, and we go into the job, and training can take sometimes a month, maybe longer, depending on where you are. A lot of times it can be longer. So how long is the training? Like if someone makes the switch to a veterinary lab, how long does does the person get trained before they can start working on their own?
I think it's very tech dependent. If you have a significant background already in human testing, for microbiology specifically, sample prep and specimen processing is the same, incubation conditions, all of those kind of little nuances are the same. The learning curve for me came into play where there are a few different animal pathogens to learn that we don't see in human medicine. For me, it it was pretty easy to make a comparison. So for example, say in humans, Haemophilus influenza could be a common respiratory sinus ear type pathogen. An equivalent in veterinary medicine might be Histophilus. So there are equivalent like organisms that you can draw that correlation to that help you learn quicker. So really training, aside from reading your policies and being signed off on your competencies, it wasn't too bad.
Yeah, it very much depends on which department you jumped into. Sarah came to us with incredible experience and years of training. So she picked up on things very quickly and was able to help with things almost instantly. But I know for me personally, even with both molecular and micro experience, I was shell shocked when I switched over to the molecular diagnostics. In human medicine, we have so many tests that are FDA approved, and here's this kit, and it pops up the result, and you don't really have much for troubleshooting. Well, most of our tests, because we are looking for very specific pathogens for different animal species, are laboratory developed. So you really end up becoming quite the expert at, your tests very, very well. So you do get very skilled. And something else for training too that I know neither of us had a whole lot was media preparation. There was a lot of media that we prepared. I think Sarah picked up on it pretty quickly, but I think that was kind of a whole new animal that neither of us had done much with.
Okay, yeah, it makes sense, and especially like in micro, the organisms are the organisms, like you say, learning some that are specific to animals, and also, I would think just right shifting your thoughts and then about so what's pathogenic versus what's normal flora, and some of those we're even familiar with, right? So we know about Pasteurella, we know about Salmonella, but then when it comes to that specific animal, it's different. It's part of their flora. So I see that. So now that we're talking about testing, so what type of testing do you perform?
So our laboratory is really well known for toxicology. We have an incredible boarded toxicologist that works here, but we also have a very wide complement of molecular diagnostic tests, different cultures, serology, virology. As Sarah mentioned, we have parasitology, and because we have pathologists and we serve our state, we also perform necropsies, which are animal autopsies. And so we're getting a lot of testing from that versus just clinical samples being sent in.
Also, a key component to our facility as a whole is rabies testing. We are mandated to do that. That's one of the primary reasons we exist, I guess, is to provide rabies testing for animals within our state.
Yes, that's written within our century code, is we must exist to perform diagnostic testing and rabies. Very good point.
Yeah, definitely, right? So so what about in micro, as far as what kind of instruments do you have over there? If anything, or is it pretty much you use the same? And I'm kind of just mentioning because I kind of asked someone that was working at SeaWorld, and then they mentioned, right, that they even had an instrument, and this is it's not an endorsement or anything, but like the Vitek, they had a Vitek and they used it, and so it just makes me curious about what kind of instruments do you use in the micro lab?
Yeah, I think we use, instruments that you'd be familiar with. We have a mass spec for the majority of our identifications. We have a reader for AST panels. We are in the midst of bringing in a Vitek, but I view this as kind of one of those great opportunities in vet med as a place where we can advocate for change and improvements. We know there's all kinds of automated and fancy instruments that we can use in human medicine and diagnostics, and they could easily be adapted for veterinary use. They just aren't yet. So that's, I guess, kind of an area, I guess, that I feel Kelli and I are a bit more vocal about. prompting some changes in that realm.
Yes, vendors, there's a lot of opportunity in veterinary microbiology and veterinary medicine in general. We're always looking for tools, and a lot of times you have to adapt, but very simple tweaks with a lot of common instruments would really be major life improvements.
Okay, so so there's the gap is very significant between what's out there for human medicine versus veterinary medicine, and that's something that needs to be worked on.
Yeah, I would mention one thing we are very acutely aware of, that we're in a very resource limited setting. Like, we are very cost conscious. So we can't throw $700 at a really fancy test panel for someone's dog. That has to be affordable for them, or for a producer who has an entire herd of animals. It's not practical in most situations to test them. So we have to be very selective and conscientious of what we're choosing for our test methods. And so then adapting that, there's a lot of opportunity for that.
Okay. So, and then I just want to ask about cultures, right? I'm definitely, still is a big part, even though in microbiology, clinical micro, a lot of, molecular, keeps making huge leaps and bounds, but we still do culture. So maybe can we talk about some of the cultures, if you do any over there, and maybe give an example of what a workup would be, based on what the animals, normal flora will be versus the pathogens, what you're expecting there.
I guess I would say we perform all the same types of cultures that you would encounter in a human lab, along with the very similar workflow. When you say you're growing a culture from a lung respiratory sample, it's still the same. You evaluate your growth, and you determine or separate your normal flora from your pathogens. Normal flora is normal flora. There's always, the alpha streps that are normal, and there's, weird types of Neisseria and Haemophilus that are normal in most of our animal species. But the kind of unique component to many of our samples is that we don't necessarily get our samples immediately after collection. Sometimes maybe an animal has been out in a pasture for a day or two before, the owner finds it and decides to send a sample in to determine a cause of death. So we wind up separating out a lot of environmental contamination, and situational bacteria that might show up from older samples, so it just adds another layer to our cultures. Not only are you determining normal flora from pathogens, but you're also separating out environmental contaminants.
Another bit that thankfully we don't experience as much in animals, think of how an animal uses their tongue and how much that can impact interpreting a wound or an eyeball. So it really makes it interesting to interpret some of that flora too. But that post-mortem overgrowth or suboptimal sample conditions it makes the interpretation more challenging sometimes.
Oh, that's very interesting. Yeah, because normally we find like that type of unusual, thinking about respiratory flora, and then we see, right, when we think about Eikenella, and we talked about, right, a human bite, or sometimes we see between, little kids, and siblings, that one will bite the other, and then you get respiratory flora, let's say on the arm or the forehead. You've seen that. So definitely you have to account for that. And then, yeah, definitely the environmental factor. It's not like us, right? We see something wrong, we go get seen right away for the most part, and so those samples are fresh, and we start working right away with them. So you have that extra challenge. And then you mentioned susceptibilities. Can you talk more about that? I mean, what do you typically do?
Go ahead, Kelli.
Okay. Yeah, susceptibility testing. We do broth microdilution primarily. We are looking more into actually picking up more Kirby-Bauer disc diffusion and e-test. We are also, as Sarah had previously mentioned, we're working on bringing a Vitek in. The antimicrobials, again, they vary by the animal species. What is approved for a companion animal is not the same as for a food animal. And part of that are regulations through the FDA on what is appropriate for a food animal to consume. There are certain antibiotics that they should never consume, such as like an aminoglycoside. So that will vary quite a bit based on those species. Both Sarah and I are involved with the Clinical and Laboratory Standards Institute on the Veterinary Antimicrobial Susceptibility Testing Committee. So we are part of the standards developing group for us. Veto1S is the analogous document that you use M100. It's analogous to M100. So we would use that similarly. And that document is an animal in itself, M100, but Veto1S has so much to learn, and there are so many little factors you have to consider when selecting the right antimicrobials for the animals. And something that differs, so we're used to getting samples directly, usually from like in your hospital system. So there's specific information you have formularies of drugs that you're testing regularly. So you have those expectations. We have to, as a laboratory, independently select which antimicrobials we're routinely going to test and provide a client. We don't know if the animal has pyelonephritis, so is it appropriate to report a fluoroquinolone and allow the clinician to have that freedom and judgment, or, versus the hospital system, which might say susceptible to these lactams or combination agents, then you don't get X, Y, and Z. So there's some of those challenges associated. So it's a very complex process. And Sarah, definitely add to that, please.
Yeah, no, to me, or susceptibility testing is really fascinating in general, right? We can tell a physician or a veterinarian a whole host of antibiotics that may or may not work in the human land. But for veterinary medicine, not only do we not have guidelines or breakpoints for all species and organism combinations, but each animal may metabolize a drug differently. And so, I mean, there's all of these different factors that are out there that we, again, may or may not have the benefit of working with a veterinary pharmacologist and a veterinarian. So, we do the best we can with the drugs that are available in veterinary medicine, and the testing options we have that encompass those drugs. But it's definitely a work in progress.
Okay, yeah. And I see that. I mean, yeah, as far as I think about it, with because the drugs that are already there and they're using human medicine, they have already their mechanism of action. So it's thinking definitely about breakpoints and how that particular animal will metabolize that and how it's different and will it be okay. So yeah, there's definitely more to learn, it seems. Is there anything else? I know that you perhaps had mentioned about any stories. I know that on veterinary medicine you deal with, right, you talked about rabies, and sometimes with animals we see, right, anthrax, we see plague. So, and do you have any stories like that, anything that you have worked with or anything to share with the audience?
Yes. So as medical laboratory scientists, we know we train to see anthrax. We train to be able to recognize Yersinia pestis and tularemia. But I had no appreciation for anthrax being endemic in North Dakota, and until working here and just how frequently it's seen. We're currently in the midst of an anthrax outbreak in the state. It's localized to a particular county. So we identified that using a PCR assay. We would have the ability to culture, but just for safety's sake, we wouldn't. We've also seen a lot of tularemia this year too, in cats primarily. Anthrax you end up seeing in cattle, or other ruminant species like goats. So there's just always something different. And I know Sarah has also experienced the joy of just pathogens you never really expect to see very often, like listeria, and just seeing that out of a sample on a routine basis is always just really interesting. There's just always something new.
Yeah, right. I mean, like you mentioned, we train for it. We get tested every year. We get this survey where we get some samples with some case studies, and the thing is, like most of us working in a lab, and gratefully so, of course, right? We probably go our whole careers without ever in the human population working with anthrax or the Yersinia pestis. We'll see that sample, and that's a good thing, right? Maybe the most we get exposed to, we get Brucella in the lab and maybe some Francisella, but that's about it. So that's like like working with it and seeing so much, it has to be an experience. And I mean, good that you work with it on a PCR. I mean, it'll be kind of like shocking, right? Seeing all those plates growing it, and that's one of the things that comes to mind. That the person that I mentioned that worked in a zoo, sometimes she said that at the beginning it was an adjustment, like working cultures, to say, from chickens and seeing so much salmonella. Like you're trained with the human population, you see that, you report, it goes through this whole process, and so it's very interesting, right? Seeing what's pathogenic somewhere, it's commensal in another species, and it's, to me, that's just very fascinating.
And
Well,
that's
it's
another
definitely,
good point with
yes.
the oh sorry, and that's another good point with the commensal Campylobacter. We expect that to be a pathogen pretty much in any human, and it can be passing through a dog at any point in time. So there's debate on what things are commensals versus actually causing disease. And right away, that makes me think of dogs and Campylobacter. Campi jejuni, no one's really debating that one, but Upsaliensis and just some of those other species, it's a really interesting field, and there's so much opportunity to learn and continue to grow and contribute to the field.
Yeah, interesting indeed. Yeah, and they create so much work in human medicine, especially in the summer. People start doing the outdoors, and maybe not with COVID, after COVID, not so much, but summertime, especially in smaller labs, it was a very busy time where you were just, especially if you had to do manually fill out the paperwork and send everything to the state. It was just some very busy times during the summer. Yeah. Well,
There,
yes.
I was going to say, I think there's another unique aspect of veterinary medicine is really this focus on One Health initiatives. I don't know if you're familiar with that, but it's basically kind of saying that, you have humans and you have animals and you have your environment, and they're all interconnected. And so what affects one is going to have an impact on the other. And I feel like it is very evident in veterinary medicine, how that idea, or how what we are doing as MLSs does impact One Health. If you have cattle that are sick, they say now we have our anthrax outbreak, right? In cattle in North Dakota. If we aren't reporting those in a timely manner, we aren't getting those results communicated both to the veterinarian, the rancher, as well as state health officials. The animals aren't being disposed of properly, which then goes on to impact, the environment, which then goes on to lead to, further anthrax outbreaks in the future. So you just really do see this whole global perspective in veterinary medicine in almost every case. You can see how, your dog having an ESBL producer could maybe cause an impact to that animal's owner, depending on hygienic practices within the home. So it's a very cool aspect and a very interesting way of following, a pathogen throughout, its life cycle.
Yeah, it does sound fascinating, and there's definitely a connection, right? And we know about how the organisms, and then the water, and so it's everything. It's connected antimicrobial resistance, and how the organisms, via plasmid, they pass from resistant to another, and there's a lot of connection. Is there anything else that either of you want to add?
I think it's covered pretty well. Definitely an excellent avenue for someone who would love to marry their love of science and the medical laboratory and veterinary medicine. There are a lot of opportunity in the field.
Okay. And you are currently hiring people?
We are not currently hiring, but we do have retirements. Just like in the medical laboratory science field, we're always just waiting for that retirement. We're hitting that. I know for me, when I was looking to join the field, my advisors were, it's a great time to get in. The biggest generation will be retiring. There's so much opportunity. And I would say we are still in the midst of that. We're losing some very experienced individuals, and eventually we'll need to be hiring. So.
Okay. Yeah, that's how, then that happens in all aspects of all areas of the lab. And certainly was for me when I came in. You have a lot of techs, they've been doing it for 20, 30 years. Very hard to get a job on first shift. So you have to kind of work the second or the third just to be in the department waiting for something. And then retirements, people start leaving, and then all these openings. And so, yeah, there are some cycles to that. Well, Sarah and Kelli, it's been great. Again, like I mentioned before off the air, I really been wanting to do this for a long time and learn about what happens in a veterinary lab and in microbiology. So this has been so great. So thank you so much for taking the time to coming into Let's Talk Micro.
Thanks for having us.
You bet.
My pleasure. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about veterinary microbiology. As always, I enjoy sharing this information with you. So always, try to bring that passion to what you do. And I hope that as we progress, as we bring more awareness about this profession of medical laboratory sciences, that we can start mitigating some of those factors. It's always very unfortunate, very sad when we experience burnout and we lose people in the profession that we need so desperately, to that. So I hope that, that can change in the future. So please bring that passion to what you do. It's so important. We do such great work. And stay tuned. Great things coming your way. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.